There is a change in the primary structure of what? Glutamate to valine in what? The protein what? What protein are we talking about? Hemoglobin. Okay. So, glutamate. Tell me something about glutamate that you know. What if I say glutamate? What are you supposed to think about in terms of what classification? It is a negatively charged amino acid. So where should that particular amino acid be on the surface of the? Oh, it should be on the surface of the protein, interacting with water, making it soluble, right? If I say valine, how would you categorize valine in terms of what category of kind of amino acid it is? Hydrophobic. That should be on the inside, hydrophobics, but now I have this hydrophobic patch on the outside of the hemoglobin. That seems not to be a good thing, and what happens is it becomes a very bad thing, okay? Because with that valine patch, or what we're going to call it is a sticky patch, it's a hydrophobic patch, okay? It allows the hemoglobins to stick together just like Legos. And they clamp together, and they clamp together, and they clamp together, and instead of hemoglobin being free to roam around inside the red blood cell, they all create this big, long rod, okay? And it connects all of them together, even to the point that when it gets to the edges of the cell, it still continues to make a rod. And it takes the round cell, and it just deforms because I have all of my amino acids, all of the proteins making one big rod. Okay. How prevalent is this? It is seen more often in the African American population, genetically transmitted. Homozygotes have sickling. About 50% of the red blood cells are sickled. Heterozygotes have the trait, usually not symptomatic, and about 1% of their red bloods are sickled. We identify a sickle cell hemoglobin by giving it the designation hemoglobin HbS, okay, to say it's sickled. We now have other hemoglobins. HbA is going to be the adult, and HbF will be what? Fetal. And we know fetal behaves differently because it has different chains. The beta chains are exchanged for the gamma chains. Now, we now know the change is a valine in place of a glutamate, but before that, we didn't. And so how did they come to the conclusion that this was the amino acid change that happened? All right? We actually now know it is a replacement in the beta chains, not the alpha chains. So 2 experiments came to the conclusion that a valine, the diseased protein, replaced a normal glutamate. Isoelectric point determination and then fingerprinting of the hemoglobin. So, isoelectric point. Oh, how does that separate? Using isoelectric point. Okay. Electrophoresis, right? That means I'm going to put a positive electrode and a negative electrode, right? And then the electrical current is going to make them move, right? Because every protein has a charge. If it's positive, it'll go to the negative. If it's negative, it'll go to the positive. But proteins have a charge only if they're at a certain pH, right? And so what we do is we have a gel that has different pHs throughout the gel, and so it'll move until it reaches a pH that is equal to its pI, making the same number of positives, same number of negatives, and it becomes neutral. It doesn't move anymore. So if I take normal hemoglobin, the adult version, a sample, runs here. If I take a disease state, right here. And if I have someone who has both traits, I can see both. Okay? Now, what we do from the origin where they start, okay, what we find is a difference. Okay? Now that difference can be measured by how many pH units there are between the resting point here and the resting point there. And the difference in PIs based upon the pH was about 0.23 pH units. What that represented was a difference, okay, of a difference in one pH unit is about 13 residues. Okay, meaning this, I went from either neutral to negative, 13 of them, or I went from positive to neutral, that kind of change. Okay, and so if I have a 0.23 change and a full unit pH change would be about 13, they concluded there could be 2 to 4 residues that were changed. Okay, now 2 to 4, not too bad. We have 4 subunits, right? If we had change in one each of them, that would have been 4. But we also had 2 versions of subunits, alpha and betas, and we could have had only one change in the betas or one change in the alphas, and that would have been 2. All right, so we got some information. Was this a lot of information? Well, it got someone published, but it didn't tell you exactly what was happening. Okay, so further experiments are needed. And what we did was we fingerprinted. Okay, so what we're going to do, we're going to mix or take the protein and break it apart. Okay, we're going to digest it into pieces. Then we're going to use electrophoresis and chromatography. All right, so you take your protein, a little bit of sample, you allow a protease, an enzyme that breaks proteins, and it breaks it into pieces. And I put the whole mixture on this, actually it's a piece of paper when they did this one. This is old school. They did electrophoresis, okay, in terms of moving, and then they did chromatography, both of which, one moves it because of size, one moves it because of its ability to adhere to the paper versus the mobile phase. What we do is we separate a very large mixture into a very large field. Okay? This is what we looked at. We saw different peptides. And at the top is the hemoglobin that's adult, and at the bottom there's a hemoglobin that is sickle cell. If you actually lay them on top of each other, all of the dots line up because they weren't changed except for one. Okay? One of the pieces was different. That means physically there was something different about that. So what we did was we cut this one out, and we cut that one out, and we did sequencing. And we said, phenylalanine, good. Alanine, good. Valine, glutamate, oh. And the next one, leucine. And when they sequenced the whole thing, there was one change. Now, by doing that sequencing, they could determine, this was not alpha subunit because alpha doesn't have this sequence. It was the beta subunit. And in the disease state, there was a valine where there was a glutamate in the original. Okay? And so that lined up to have 2 changes for the hemoglobin, which then was supported by the other experimentation. Okay, in essence, just some details here. They used the enzyme trypsin, which broke the hemoglobin into 28 pieces, and so they had to track 28 pieces in that. Only one of them were different, and then sequenced. Okay, now why in the world does this one valine have a problem? All right, let me show you what happens. Okay, so here is a detailed version of hemoglobin. You see it? Okay, what am I representing in this designation here? They're what? What do you see? 4, right? 4 subunits. Okay, so there are 4 proteins working together as hemoglobin, 2 alphas, 2 betas. In the oxy form, no big deal. In the deoxy form, there are in the alpha subunits a hydrophobic pocket that opens up. Okay, Now, if I had a lot of oxys, none of the hemoglobin stick together. If I have a lot of the deoxys, that hydrophobic pocket is on the inside. So if I try to match them up and put them together, well, I got hydrophobic pocket on the inside and a hydrophobic pocket on the inside. There's nothing to interact because they're inside. And so if I get a lot of this together, nothing matches together. If I've got a lot of this together, nothing binds together. Okay? Here is the sickle cell. Having one valine out there, okay, if I have a lot of this in the oxy form, that valine doesn't fit together very well, and so they don't bind together. But here is the bad part. If I've got this version, the deoxy form, do you see how if I had another one, it fits together? That this pocket would be right here, and that valine sticking out would fit right there. And so the structures in the deoxy form fit together and bond. Because there is naturally a hydrophobic pocket that opens up in the deoxy form, okay, normally there's nothing to stick into it, so no aggregation occurs. But because of this sticking out, the aggregation can occur and they can bond together. Right? And so the sticky patch binds into that nonpolar pocket. Okay. All it is van der Waals forces. But if I've got those van der Waals forces that are perfectly aligned and they come together, they will stick and form a fiber inside that red blood cell and distort it. So, one change in one amino acid caused a disease, an anemic disease that has issues. Now, it's really bad because of the deoxy form is what happens. Okay? It's buried. I have lots and lots of slides that says what I just said. Okay? Here's the vicious cycle. All right, red blood cell, nice, round, flows through the veins and the arteries and the capillaries. But when it becomes deoxy, it can sickle. It can get trapped. Okay? Another one comes up and it can't move, but I lack oxygen, so it sickles and lets out oxygen. And I can create this barrier such that on this side, I'm letting out lots and lots of oxygen. On this side, I lack oxygen, and so you can have cellular damage because it's lacking oxygen. The only way, if by chance a sickle cell gets up to the lungs, it will come back to a perfect round shape because the oxy form doesn't bind together. The oxygen can cause these to come and break apart. So if it gets up in the lungs, it's fine. But if any sickling happens far away from the lungs, then we can have lots and lots of blockages. Okay. Oh my goodness. Hopefully I need to warn anybody who might have epileptic shock here. What we're looking at is the next concept. Okay. We're looking at a bilayer. Okay. So I like this, other than it just is weird. I like this slide because, oh, good night. Okay, let's stop it. All right. Because you see all the water on this side, and you see all the water on that side, and you see all this hydrophobic in the middle. Okay? And so you've got lots and lots of hydrophobic nature with the fats that are here. There's a polar edge on this side, and there's a polar edge on this side. This is the membrane. Water on this side can't get through because I've got lots and lots of hydrophobic. And so I create one compartment on this side and one compartment on that side. Polar molecules are impermeable to this barrier by the way I look at it just right here. And so we have lots and lots of membranes. But here's the problem. If a membrane doesn't allow anything to move through, what would happen? If you can't move anything through, you would die. That's right. You have to have function. All right? The cells have to take in nutrients. Okay? Oxygen, food, right? And it's going to produce waste. And so it's got to get that waste out of the cell. Now, what is important about the membrane is the control of what we bring in and what we let out. Each cell has a membrane that controls what it does. Each organelle has a membrane that controls what it does. And so there are different compartments by which we control it, all the function. Now, what we're going to have to do is, through that bilayer, that lipid bilayer, how in the world do we control the movement of things through there? Okay? We must control that, and that is done through proteins that are integral or actually embedded in that bilayer. Okay? They will transport all of the stuff, ions, molecules, whatever, we need through that membrane. There are two types, main types of transport proteins. One are called channels, and one are called pumps. The channels are passive, the pumps are active. Okay, so what in the world is the difference between passive and active? Energy. How much energy you put there. Okay. One, well, doesn't require the breakage of energy, most of which is directly ATP. We break the bond of ATP to move something across the membrane. Channels are usually passive, means that if I open up the channel, things are going to move through. I don't have to invest energy to do that. Now, the pumps are not going to be exclusively ATP. There are other energy sources that we can use, and I'll show that to you in a little bit. Channels don't require the input directly of energy. All right, so a couple of things. There is a place in time in which a molecule can get through the membrane, all right? And that's simple diffusion. What happens in simple diffusion is this. The molecule has to be very hydrophobic. If it's hydrophobic, it can get inside that membrane and go through the membrane. There's a problem with something very hydrophobic. It doesn't like to be in the water, so it has to be carried to the membrane by something. It can get through the membrane by itself, but then it has to be picked up by something and carried inside. Okay? And so, how do I get through? All right? Well, how fast it goes versus how concentrated it is. If I put lots of concentration on the outside, the rate at which it goes through is linear. Okay? And so, the more you put, the faster it gets. All right. You have a passive transport. What's important about this? Okay, the passive transport takes a large concentration and allows it to move down the gradient. That's how passive works. It goes down a gradient. All right. Now, two different ways. One, passive transport channel is a way that I can do it. It's a protein that basically does this. It's closed and opens up a hole. Okay? It opens up a hole, then things should be able to move down, and things will move down. Okay? The channel and the rate. The larger the difference you have on either side, once you open up that hole, the faster the rate will be. And so it is a linear. There's also a passive transport carrier. It's a little bit different. Instead of going through the membrane like this, like closed and open, okay, it does this. It opens itself to the outside. Okay? There's a lot of stuff outside. It will bind it. When it binds it, it will open to the inside and stuff will fall through. Okay, once it lets go, it changes its shape and opens to the other side. So it works like this in terms of the protein's function. Okay? And it allows passive transport. Now, in that, there is a rate at which it binds and a rate at which it lets go. And then it has to go back and forth. As I put more and more difference across that membrane, it does work the more you get, the faster it goes linearly, but then all of a sudden, it gets to a maximum rate. Okay? Why does it maximize out there? It becomes the fastest it can go is the rate at which it can transition. Okay? You put a big enough gradient, it should go really, really fast. And so, but then it's limited by how fast it can work. And that's as fast as you can go, even if there's a larger gradient. Here, if you just have a hole, you got a big difference. It'll go very, very quickly. But here, I actually have to bind to it. All right. In the case of the active transport, here are the big pieces about it. What happens is, in active transport, a small concentration can move to a large concentration. That is energetically unfavorable. This is why we have to input energy. I take a small and put it into a large concentration. To do that, I will use some energy, in this case, normally ATP. And in almost all of these cases, you actually have to bind to the entity, whatever you're transporting, and move it from one side to the other. And so the rate at which it transports has a saturation point because it can only go as fast as it actually can convert. And so we can do a lot of looks about how things are being transported and put them into 4 different categories. Okay, passive, down a gradient. Okay, and so from high, and when I open it up, it goes to low. Okay, how does this actually help, or how does the protein actually help? Okay, here's an ion surrounded by water. That means it is polar. Here is an ion surrounded by waters. It's happy. Here is something inside the hydrophobic core. How happy is that? Very unhappy. Okay? Energetically, it takes a lot of energy to do that. So much so that it's not going to happen even if I heat up the solution, although we don't regulate our bodies by heating it up and cooling it down. What we're going to do is, this is surrounded by polar, this is surrounded by polar. When this molecule is inside this protein, all of the R groups around this are going to be polar. So it looks like it's in water, it looks like it's in a polar environment, and it looks like it's in a polar environment, only on the inside. On the outside of this protein, it's going to be hydrophobic because it has to interact with the hydrophobic. Remember how I told you proteins fold? What's on the inside when it folds? Hydrophobic. That is in globular proteins that are floating around. Here, what's on the inside of this protein? Polar. What's on the outside? Hydrophobic, because this lives in a hydrophobic environment and has a polar core to interact with something that wants to interact with polar. So this integral protein is exactly opposite of all the globular proteins that you see floating around in the cytosol. Okay. Now, there's another way that we can define things in terms of how it moves things across the membrane. Okay, uniport, got that. I move one thing across the membrane. That's no problem. Symport, what's happening with that? 2 things come together in the same direction, right? Antiport, 2 things are moved, right? But one in one direction, one in the other direction. So in any transporter, it is fair for me to ask, can you categorize if it's uniport, symport, or antiport? Okay. All right. Again, thousands of transporters will show you about 12. Right? And then you have to make allusions for the rest of it. Here is one transporter called aquaporin. It is passive transport. Okay. It is functional as a tetrameric protein, and it has 4 little channels. Okay. And you can see water. Here I've got lots of alpha helix. But you see this little trail right here? That's the opening. Okay. You can look at it from the top. Here's the trail of waters. They go right through. Okay, it allows water to pass. The rate at which it passes, 5 e to the 8th per second. Okay, make that a real number for me. What is 5 e to the 8th? Okay, if I gave you 5 e to the 8th dollars, okay, how happy would you be? What would that number be? You'd be very happy? Good. If I gave you 5 e to the 8 pennies, how happy would you be? I'd still be pretty happy. Okay? 5 e to the 6th, what number is that? 5 million. So that's 500 million waters per second. Okay? That's booking it. Right? That's a lot of water moving through that. Now, what actually forces this? Osmotic pressure. That is the difference between the two, and this facilitates. Here's the problem. I told you that the liquid bilayer is a barrier to polar things. The only thing that can actually leak through is water, depending upon how big the osmotic pressure, but the aquaporin are there to facilitate that. Water is the only polar molecule that can actually make it through the membrane. Now, what we know is it's only a 3.8 angstrom opening, and it allows just enough space for a single water molecule to move through. Okay? And so what has to happen is, remember, this water is coming through, but it's got to interact with polar residues. So it has to be close enough to the sides to interact to make sure that it is interacting with polar residues as it goes through. So it is perfectly aligned and perfectly shaped to allow only one water to make interactions on both sides of the protein as it falls all the way through. Okay? The next one that is mostly taught is glucose transporter 1, GLUT1. Okay, when I say GLUT1, what does that mean is probably going to happen? GLUT2, GLUT3, there's lots of glucose transporters. Okay, so looking at this glucose transporter, okay, things that we know about this. Okay, we've got a pretty good structure. Take a look at the hydrophobic, polar, and the charged residues. Okay, here is where the membrane happens. Here's the membrane, and these are helical bundles that fit inside the membrane. All right, here is in the solution. Look in the solution. You've got lots of polar residues because it's in water. Over here, this is in solution. There's a lot of polar residues because it's in water. Here is lipids, right? And here is nonpolar, lipid and nonpolar. That's great. But what in the world do you see in all of these alpha helix? Lots of what? Polar residues. Polar residues are not supposed to be in the hydrophobic area. All right? And so it looks like I'm lying to you straight up, but I am not, because this is what happens. When an alpha helix folds around, okay, if we put the residues in the right positions, look, in the 2, 6, 3, and 7 position, I have all polar residues on one side, and in the 1, 4, and 5 positions, I have polar residues on one side. And when they bundle together, take a look at this. Polar, 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 polar. And on the outside, all hydrophobic. So on this side, it's interacting with the membrane, and it's embedded in the membrane. But inside, it creates a hole that has a lot of polar residues, so that glucose, which is very polar, why is glucose polar? I know it's sweet, but why is it polar? Because of oxygen and hydrogen. And when you get oxygen and hydrogen bound together, you get a special polar bond called hydrogen bonds. And how many hydrogen bonds does glucose have? Ooh. You've seen glucose all your life. But now I'm asking you the chemistry behind glucose. There are 6 of them. Very, very polar. Got it? I I got it. Okay. And so, if you take a look at how these alpha helices line up, we can create that polar channel for glucose to go through. Okay. Now, here's how glucose transporter 1 works. All right. And so, we've got a circle. Where do you want to start? 1. Yeah, you can start at 1, but if it's a circle, you can start anywhere. Okay. So here is the outside. Here is the inside of the cell. Glucose on the outside of the cell. I have just eaten my Snickers. Okay. Glucose fits inside a binding pocket that is perfectly aligned for glucose. Has a high affinity for glucose, and it binds in. Okay. When that glucose binds, what happens is small little conformational changes in the protein occur. And so I twisted a little bit. Some of the bonds with the hydrogen bonds of glucose make things move, and a little bit movement here, a little bit movement there, causes a large conformational change on the outside. And so I open to the inside. Now, this pocket doesn't like and doesn't bind to glucose very highly. It has a low affinity. So if it has a low affinity, then this thing floats in. But as soon as this thing falls apart, what was making this shape possible also leaves. So what happens is it reverts back to its original shape in which it can bind another glucose. And it does the bind, let go, bind, let go. Now what's important, this first one, this bind, has a high affinity for glucose. When it changes, it has a low affinity for glucose. And high to low, high to low. Okay? And it will move down the gradient. The kinetics saturate. Okay? Because at the beginning, if you give it just a little bit of glucose, it'll go at a certain rate. If you double the amount of glucose, it'll double. But there is a point at which you aren't measuring the difference in concentration anymore. There's a point at which you're measuring how fast this can flip, the rate of the flip. Okay? Now, in previous teachings, there's something called the Michaelis-Menten-like. You don't know what that is yet. And so just wait till the next chapter. It'll all become apparent. So that nomenclature you may not understand at this moment in time. A pump works differently in terms of there has to be some sort of energy placed into the system. Now, most of the energy comes from ATP. And what we're doing is we're taking a low concentration to a high concentration and we're moving it against the gradient. ATP is not the only power source. I can use an ion or proton gradient as the power source. I can even use light as a power source. We'll see that in other places. Okay, and we go against the gradient. Okay, any gradient that is created is a potential energy source. So what I'm doing is I am increasing or transforming the energy from, let's say, ATP into a different energy source, a gradient, a difference of ions across a membrane. Okay. There are two types, okay? Active primary transport is I break ATP directly and I take something from a low concentration to a high concentration. That's our traditional, that's our classic. I also said a gradient could use this. And so I can move something from a low concentration to a high concentration if I take something from a high concentration and couple it to a low concentration. What kind of transport is this? Which one? I couldn't. Is it facilitated? It is facilitated, but of the three that I asked you, Symport. Okay. It is Symport. All right. In this is the energy source. But this energy source is a secondary, okay, active transport, because this energy source had to have been created at some point in time, usually with the breakdown of ATP. Okay, so I can take something from low to high, and I'm using this energy source, which was derived from a primary source. So you can talk about primary and secondary active transport. But yes, they are facilitated transports. Okay. All right. ATP-driven pumps. There are 4 types. Okay. We're going to show you a few of them. The most common one that we look at is the P-type transporter. We're going to look at the ATPase transporters, and then the ABC transporters. Those are 2 of the most often seen types. Okay, a P-type ATPase. All right, kind of interesting. One way to classify it: phosphate is the byproduct of ATP being broken down to ATP and PI. That's the PI. Vanadate is a homolog, and what happens with a P-type pump, you can tell if it's a P-type pump because vanadate will inhibit, will stop the pump. And so that's easy to identify by using that. What also happens in a P-type pump is this. At some point in time, the protein, the pump itself, gets phosphorylated. Okay, an aspartate residue picks up an ATP, reacts with an ATP, and a phosphate is transferred to it. So it becomes phosphorylated in the midst of the processing. Most of them have at least one large single polypeptide that will span the region. Some of them do have subunits. Okay, here's the most studied. The sodium potassium ATPase. Okay, we're going to come back to this quite often. In all cells, the concentration gradient of the sodium and the potassium is this way. On the outside of the cell, there's a very large sodium gradient. There's very large concentration of sodium. And on the inside of the cell, there's a very high concentration of potassium. Okay? There is a differential though. Okay. Inside the cell, there are less ions in terms of the potassium and sodium, and so it is negatively charged on the inside of the cell. It is positively charged on the outside of the cell because we're going to transport these differentially. We're going to put more positives on the outside than we put negatives on the positives on the inside. All right. And what we create is an electrochemical gradient across membranes. Okay. The concentrations are maintained by the sodium-potassium ATPase, or the sodium-potassium pump, and it's going to move these ions. The transport happens this way: 3 sodiums out for every 2 potassiums in. Okay, and that will happen for every ATP that is broken. So 1 ATP moves 3 out, 2 in. All right, we have not a very great detailed crystal structure of the protein, but we do know a couple things about it. It consists of 2 alpha subunits that are 112 kilodaltons, 2 beta, which are 35 kilodaltons, and it's a tetramer, 2 alpha, 2 beta. It has about 8 trans-membrane helix that create the core where things move through. We know that the ATP binds on the cytosolic face of the pump, and we know that there are some sugars that are faced outside the cell. All right. Now, when we look at the sodium-potassium ATPase, we're going to also understand the calcium ATPase because their mechanisms are similar. All right. The action of this will cause both the concentration and electrical gradients. All right, here it is. So, on the inside, we want sodium to be low here, and so when the pump is open to the inside of the cell, it has 3 very high affinity pockets for sodium. 3 will bind. Okay? When that occurs, ATP will then be facilitated in such a way that the ATP will be broken down consequently, and aspartate will be phosphorylated. Now, when we phosphorylate one amino acid, we really change its structure and its charge. And so what happens is this phosphate allows a conformational change to occur. And it goes from the inside to the outside. Now, these pockets, what do you think about their affinity for sodium? It was high on the inside, it's now what? Very low. So they fall out. Okay? And it has to be extremely low because there's lots and lots of sodium. So it has to go from really high to very, very low. And the sodiums move out. Now, what has happened in a consequence? I have 3 very low affinity sites for sodium. What have I created when I opened up? 2 very high affinity sites for potassium. And potassium's relatively low concentration here, but it'll bind because these are high affinity sites. When the potassium gets in here, 2 things occur. First, the potassium causes the protein to shift and allows the phosphate to be dephosphorylated or be broken off. When that is broken off, which is what's kept it open to the outside, once it breaks off, it flips to the inside. What happens to these affinities? They drop. And so potassium now floats away. And it has to go really low because remember, potassium is pretty high on the inside, so it has to go really, really low. And so back and forth, back and forth, back and forth. Every time I break this, every time I transport 5 ions, it costs 1 ATP. Okay? If by chance you took a day and you sat on your couch and you streamed Netflix all day long, okay, and you didn't move, the single greatest amount of ATP that would have been used in your body was this enzyme right here. Okay? The other place that you could outperform this is muscle movement. But if you're just a couch potato, not using hardly any muscle movement, then muscle could be the highest utilization of ATP. But this one is moving all the time. All right. The sodium potassium ATPase, wonderful protein, okay, does a lot of work. Interestingly enough, okay, we use an inhibitor of the pot called obane, okay, and obane is actually an interesting little molecule. It is the poison on a poison dart. It can be found on certain frogs. And so it's an inhibitor, okay? And it will bind to the sodium potassium ATPase and it will stop it. Now, here's the problem. To have a gradient of sodium potassium across a membrane is really important, especially for certain molecules like the nerves, because your nerve signal is the movement of sodium potassium across a membrane. If you stop the pump, you stop nerves. Okay, if you stop nerves, then you can stop all sorts of things like breathing, things like that. And so if you have a lot of this in your body, not good. It's a neurotoxin. It's a toxin to your neurology because you stop it from working. All right, but we use this as a medicine. How do we use this as a medicine? We give very, very low dosages. Okay? Now, why in the world will we do this? If we inhibit the sodium-potassium pump just a little bit, in one tissue type, there's a benefit. Okay? What happens is sodium starts to build up inside the cell. Okay? In the cardiac muscle, another pump takes over called the sodium-calcium pump. What it's going to exchange is sodium out, calcium in. Okay? This can be used, okay? It isn't what naturally we want to use. Use it a little bit, but not a lot. But what can happen is, in the heart muscle, there can be higher concentrations of calcium built up inside the heart muscle. Muscle contraction is directly related to the calcium concentration, and so if I increase the calcium concentration inside of the heart muscles, the contractions can be harder than normal. Okay? And so if people are having heart conditions that need to have much harder pumping than they normally do, we use a little bit of this to make sure more calcium is in the heart. And so it's a risk reward. We stop a little bit of the potassium, sodium potassium ATPase around the entire body, so it doesn't work as efficiently, but it gives us a benefit in the heart muscle. Okay? Poison yourself a little bit so your heart will beat. Okay? Is it necessarily great? Well, it's the difference between alive and dead, yes. Okay? But with every case in which you use a medication, there is risk and there is reward. And it depends upon where you lie in what you use. And so some people are treated with very risky things because the benefit may be that they are able to live, whereas without that, they aren't. And you would not give this to somebody who doesn't have that condition because it would be very detrimental to them. And so it's always a risk reward.